So similarly to the GMO studies that we carry on previously, we get engaged in this effort with several other scientists to check for mainly the off-types in the technology. So we are looking at the sites that also are modified by the technique. And in addition, we will try to develop a traceability protocol to make sure that we can identify these organisms or varieties or animals that came about from the use of this technology that is gene editing. So this is the main goals of this project, this effort. I guess involve first the Norway, Brazil, Lebanon, Austria, I guess England, Germany? In Germany so far. Well, I think there is many difference between the previous technology that was the genetic transformation and these new technologies or protocols to change the DNA allow additional ways to change DNA in comparison with the genetic transformation for the GMOs that was used. In fact, you can reach the target, the DNA target, or one sequence in the DNA that was not possible with the recombinant DNA technology. However, up to now, there is no report that you can change just the target, the DNA targets. So the reports, the scientific reports related to the use of this technology provides the main scientific consensus now. The technology also can reach other sites, that is a non-target sites. So based on this issue or in these results, we are dealing to start to do ways to identify these products that arise from this technology, but also to design some ways to check for the risk analysis or the effects of what this modification can provide to the organism itself, but also in the environment or the human health. This will be a kind to be used or developed in the future, but we are thinking about in these issues right now. And the second main approach that we are thinking is how we can influence the legislation that are current in our country regarding to release the products that arise from these technologies. So we have a law that is included this gene editing technology as GMO, because the definition in our law covers any genetic modification caused or done in DNA or RNA sequence of one organism. So it doesn't matter which is the technology or the protocol. However, the current members of our biosafety committee think that these technologies based on CRISPR no longer can be compared with the RNA recombinant technology or genetic transformation. Thus, they believe that the products that arise from the gene editing no longer should be classified as GMO. Thus, they don't need to follow our rules that requires risk analysis and several studies in our environment and in animals and microorganisms and so on. The Brazilian law about biosafety of GMOs is based on the fact that the modification in the genome through the recombination DNA technology, then any modification that is done on DNA or RNA should be assigned as GMO, genetically modified organism. So gene edition is like a modification in DNA, mainly in DNA, and also can be assigned also as GMO based on our law. However, the biosafety committee members believes that the gene edition cannot be classified as the same way that the modification provided by recombinant DNA technology. So they also decide to consider the products of these gene edition techniques as a kind of new breeding technologies. So far, we are doing research to provide that the modification in a target, also the protocol that is allowed the modification in one site in one target also will cause modification in other targets, in other parts of the genome. Since we don't know what's happened with the modification, these several modifications that the protocol provokes, so we should consider these products arising from the CRISPR technologies or gene edition in the same way that we consider GMOs, and also we try to convince our authorities to consider as such, and also require some risk analysis as we require for GMOs. But we don't know if we will be successful or not. Similarly to the GMOs era, previously to the huge commercializations, there are 3 or 4 mainly topics regarding to that. What's happening with GMO? They decide to do a kind of agronomic studies or check if the new variety will become a weed or not, or something like that. So they carry on some research studies that will not indicate any risk or something like that. This happens with some articles, some studies, that they do some checking or some traceability, in the sense that we're looking for, with one technique that just look for similar sequence in DNA. So then maybe are not changed like the target site. So say is a kind of partial tentative to look in all genome. So they also don't find too many side effects of targets. The second issue is that most of the studies try to optimize the protocol to develop a modified organism. So not in the sense that looking for effects on this of the technology. The third issue is that the people that were behind, so the labs that were behind the GMO research or development now wants to be in place or to provide some development, some products. So there are many small industries and labs across European countries that they are using these gene edition technologies to develop products. So this didn't happen with the GMO because only big companies try to develop. And the last issue is that we don't know or we know very little, in other words. We do have one study that was published maybe 5 or 6 years ago in mice. That was a huge number of targets, sites that were reached by the same protocol that reached the target site. And also this is a classic study that showed several effects on the organisms. However, in plants we don't have too many studies. So, and we don't know how many sites are changed across the genome of the one plant. And also we need to study effects on the physiology, on the biochemistry, on the genetics that we didn't carry out this kind of studies with GMO in the sense to looking for the entire genome. So contrary to GMOs that can insert one or few transgene or DNA fragments, this technology can reach many sites in the genome. And so we don't have even the notion of the expertise to expect how many sites will be affected with one gene edition delivery. So I guess it's too early to say that we have effects on phenotype or in the genotype and will be the magnitude of the effects in each site. So we don't have this kind of experience yet. In my opinion, it's too early to say something. But since it's a modification, we can also take in consideration that if the DNA sequence that is inside one plant or one animal is there for millions of years together, they are the rate of evolution is there, so the sequence are together for a long time. So if we have many modifications in one genome, probably you can expect a kind of provocation in order to redefine the evolution of this organism. So we don't know about if the adaptation will be similar, or if the adaptation of this new organism will require a new environment, or if a particular site that was changed will be provoke a very huge effect, phenotype effect or not. So we don't have this kind of experience. this is another issue. Well, it's cheaper, in fact, but it's require a new kind of development, like the technology. If you try to avoid the genetic transformation, because you can combine CRISPRs with genetic transformation. So if you would like to avoid to deliver big piece of DNA, so we need to have a kind of direct delivery of the CRISPR system inside the nucleus. So to do that, you need to have a protoplast. To regenerate protoplast is not so simple for all the species. So in fact, yes, it is easy, it is cheaper, but it will require new developments in cell biology, I should say. So we are facing this trouble with this protoplast regeneration. So. Gene edition can reach almost every species. Instead, the recombinant DNA technology was, well, you can do also, but it is hard and it's more difficult, it's more expensive. And also, in the case of genetic transformation, you transform one cell, you need to regenerate a plant, like a CRISPR also. But the genetic transformation usually could cause several damage or damage that is too bigger for this regeneration of the cells. So then CRISPR technology will make not so or do not deliver a big fragment of DNA. So it will be changed that maybe cell can accommodate more than a transgene, a big transgene. Of course, because, we are dealing now as a, so our thinking is about one generation, one human generation that's 25 years, something like that. So in the evolution time, the things will be very different. So if we start to release this variety that are modified by gene edition, this variety can cross with the other varieties or the wild types or the other ancestors. And maybe there is back crossing and other crossing. And if we deliver several varieties, these several varieties can cross each other or can cross with each other. So we can accumulate in the should say landraces or all the varieties, many modifications. And if you have many modifications, we can change the course of these new types. And also we can provoke some speciation in future. So these types will be so different with the others that will provocate some very distinct pathway of the evolution in comparison with previous one. And in addition, we have one phenomena that we will not pay too much attention. That is the horizontal transference. So in the evolution time, there are many organisms that have human genes, and also there are microorganisms that has some plant sequences and vice versa, bacteria, and plants that have some bacterial sequence. So in the long run, of course, these genetic modifications can be also be spread not only by vertical transference or gene flow, but also by horizontal transference, as bacteria, as is usual in bacteria, in archaea, mainly in archaea bacteria. Look, in the long run, in my life experience, I learned that. So I learned that one agronomist can control disease, or a pest, or in fact, so the human beings cannot control nature. So they try to. Every new technology is linked to the control for something. We, so far, as human society, we were unable to control even a single disease up to now, or a single pest. Up to now, none. And these people, so they very narrow thinking about one. They think that DNA is the mainly molecule that controls life. I think this argument is totally upside down. I should say that is, in my opinion, we cannot control everything. Why? Because DNA, as I think it was, later on I remember the biology thinker, but the DNA is mainly a dead molecule, because the DNA does nothing. The unique feature of the main features of DNA are, so carries some information, but the DNA itself does nothing about this information, just carry. DNA allow modification, so it's not to DNA control the modification that happens in this, in any base. DNA must be replicated by the cells. The natural selection will control the modification that happens in DNA. So the main issue in biology is the cell. So any thinker or philosopher in biology that take the DNA as a base, I guess they will not survive as a hypothesis or recognition, because this will be not happens in future. So they did the same with when the recombinant DNA technology was developed. People believe that the scientists will have only two limitations: find the sequence that they need to do some first. The imagination, so to do one thing, and find the sequence to develop the new creature that. In fact, 30 years after, we have a few modifications, so the limitation is more wider than the preview in that period. And also with this technology, again, I think DNA is a remarkable molecule that you can do everything with DNA, and its continuity as DNA. Say, in other words, you can boil DNA, the double strand can separate the other, but they can rejoin itself and continue with the same genetic information. And we can build synthetic molecular DNA. So DNA, in terms of biology, is almost a dead molecule in the sense that the function or the make something. One enzyme make one bond between molecules or atoms, but the DNA does nothing. So the cell does everything to replicate the DNA, to translate, to transcription the information, and do everything else. So of course we can, if you can change the sequence, we will provide new traits, new features of the organism. Okay, but this is not the same way that you can conclude that you control life or evolution, because this modification usually have pleiotropic effects. As we did one study, Carol did the study. So the transgenic soybean, the transgene that was inserted in the soybean to make the soybean resistant to herbicide, provides also effects on the metabolic pathway in that affects the relationship between soybeans and microorganisms. Then soybean plants became more susceptible to the disease. So this is a kind of pleiotropic effect. So by making one modification, you do not control life. In fact, it's the other way around. So you make some conditions or alter some other traits that you lose totally the control of the function or the objective of the design or the protocol. So the output will be totally different of your plans. So the another issue that is linked to this one is that these people have this Mendelian view of genetics. For Mendel, looking for several genes that were far apart, control different traits, okay. But usually the genes one affect another. 98% at least of the genes interact each other within a cell. So then one modification, maybe in 2% of the genes, okay, you have a different phenotype. But for most of the traits or the characteristic of one organism is affected by multiple genes. So one modification cannot drive the evolution. Of course, you can provide another variation for the evolution to work with. But control, we're far away from the control evolution, so far. Yeah, these new technologies, the gene edition, also eles provide a way to not only to cut the DNA and allow the DNA do the repair or change one base or another, but also allow to insert some DNA fragments. This is the way that they produce the gene driver. Say they introduce a sequence. Once this sequence is in the genome of one, like in a diploid organism, if you have a diploid organism, just one sequence will convert another sequence in the other, or in the same locus, will change the sequence of the homologous chromosomes, the locus on the homologous chromosomes. So by doing that, you can impose to the population one trait that will be, as you said, will be exponentially spreading over the population very quick. So they believe that doing that they will control, again the word is control, a pest, like if you insert a gene for a male organism or a male insect, all of them will be male soon, and then they will not reproduce and you get rid of this. In fact, we have one experience that is not with CRISPR technology, but with recombinant DNA technology that was the mosquito, the Oxitec mosquito, that was quite similar in the fact that they try to kill one of the sex, but the percentage of the individuals that was killed was not 100%. Why not? It's because of the genetic background. The genetic background, I say the other genes, because if you have some insects that cross each other, they have a different genetic background. So there is some pleiotropic effects among genes. So maybe not all of them will be male. So if not only they will behave as a male, I should say, you have a room for crossing, and this will be also not only spread to the wild population, but also will not kill all the insects that cause one. This is one of the issues. The second issue that this cancer is under the evolutionary pressure. Thus, this sequence can be mutated, can be affected by epigenetic effects like methylation or something. In other words, they are not free of the evolutionary pressure on the sequence. And some of them also can cross with the other individuals. And usually there are many bacteria that are attached to these insects. And some of them also exchange some genetic material. So, in other words, we are not sure where this genetic modification or the fate of this new, so where they will be, where they are going, who will be the limits of this modification. So we cannot control what will happen. And also, as far as we have experience, not with CRISPR gene drive itself, but with similar technologies, they are not 100% efficient. So, in fact, I think the gene driver is a way to spread some genetic modification and without any control. This, in fact, we have no way to control. This is worse than to release one variety, because one variety produces seeds, you can take the seeds. But insects, they use insects. There is no way to recall an insect or do something else. So this is worse than release one variety. And this is, since we don't know what will happen in future, in different ways that this modification can be spread, so how we can forecast or take some measure to avoid some possible adverse effect in nature. Well, usually we try to figure out a single solution for a very huge trouble. This is the way that the pesticides were introduced and GMO was introduced and everything was introduced. Usually huge problems needed to have a kind of multiple ways or multiple solutions, simultaneous solutions. The main issue could be the lack of holistic approach to treat a huge trouble. And of course, when we think very narrow in the sense that the power of the technology, only in the power, okay, maybe the people are right. So this is a way to eliminate the mice in the Galapagos Island. However, this is a kind of a network that exists in the island. Why there is so many mice? It's because something so in the web of in the net web over there for food, maybe you don't have the predators in all the steps or the phase. Thus, we may introduce one predator. I don't know if they want to. This is one of another solution. But in addition, we have a ways to, since the island is not so big, we could use other approach for animals. What I'm saying is that nowadays, none of the single technology that we use include vaccines in humans. That that is a very efficient solution for several diseases, but no longer like in flu. So the vaccine for flu is not a solution like other disease. So mice has a huge ability to survive with with very stress conditions, and including for limiting food, water, and stuff. I guess any big solution, any solution for a big problem must require a holistic approach. In the holistic approach, we take care of several simultaneous solutions or measures, and also to check for which one, and you can adopt the precautionary approach simultaneously. Then you will look for also the risk of each of the measure that you can use some measure that has some kind of risk, but not only one measure for save you one big trouble. It can maybe be so. Maybe some the modification in some other parts of the genes can provide the mice more fitness, more progenies, reproductive success higher than other mice, or because and again the genetic background, the mice are different. So one modification in the same modification in different genetic backgrounds will provide different effects. It's like medicine in humans. It's because our immunological system is influenced or modulated by our microbiome, the gastrointestinal microbiome. So if one modification is subject also to these epigenetic effects, because in the pleiotropic effects of other genes. Here in Brazil we have this corn varieties that carry these Bt genes or genes for toxins, some of them are more susceptible to our insects than others because the genetic background. So then this assumption that all the mice will be the same phenotype, scientifically is not true. It's impossible. Unless you have a pure line, like you can have in plants, a pure line. So then you have just one, you have a genetic composition, or I should say, all the individuals in the same line share the same genome. Okay, the modification, then the phenotype will be the same for all. This happens only if you have a uniform genotype. This is the reason that we carry some studies in one crustacea that is the there is one crustacea that is has a kind of asexual reproduction. So all progeny has the same genotype. So the genetic modification will have provide the same phenotype for the organism in a pure line. Otherwise, can have different phenotype. And this is the reason that the efficiency usually is not 100%. And you know, the progeny can inherit if there is a crossing over in the middle of the CRISPR, you can transfer just the gene that produces the enzyme and not the other part. So you will do other modification in the one of the progeny, not in the other. Yes. So this is the reason. This is the reason that in the no, it's okay. This is the reason that in the species that have sexual dimorphism, they behave like that. So the progeny will have a lot. So it's the progeny are recombinants in comparison to the parents, and so I don't believe that this a single solution can take care of a huge problem. Mm-hmm. So far, we, as a human society, we didn't have any example of this successful control of one huge problem. We need to recognize that vaccine was a good example, but in short time, as we can see now, that the same infection agent is coming back with another genotypes, with a different genotype, so they are arising once in a while in the human population. Yeah, and also we have one issue that we use in plant breeding. If you cross two plants like that, one plant has one mutation in gene A and another plant has a mutation in gene B, when you cross, you have a double mutant. So you have a totally different genotype and totally different phenotype. If one mutation happens in one of these mice, another mutation in another part of the genome of another mice, they cross, they will have, it will be a recombinant progeny. So this recombinant progeny has more fitness, or fitness will be lower. But both can happen in nature. As you mentioned, which is the size of the progeny of a couple of mice? Can you think across time, like 10 years, 20 years? So, and what's happening in Brazil if you compare with GMO technology? So you insert a gene for the corn and soybean and so on to resist one commercial doses of herbicide. But then the other plants acquire resistance to these herbicides. So now even though 10 times a commercial dose of the herbicide can kill the weeds that we call weeds. So this can be happen with the mice as well. So no longer with this maybe the genetic modification will cause some damage to the mice. As the biotechnology is a kind of area that someone calls science, someone calls technology, but this is the stamp that these guys use it to be in one area, to find a job, to take one idea, to go through, develop one organism. Because they think only in this narrow base that if you change the DNA, you solve problems. So we still, again, these guys are product of the central dogma of biology or the genetic. If you don't be independent of this thinking, you go through this solution, because you think the DNA is everything. So then you put in your mind that, look, I will change this, that I will solve the problem. So as I mentioned previously, these guys were not faced, I think the education system, the university or the research groups or the research labs, they don't use the holistic approach like the people in genome course use, to looking for one problem and then the solution for a problem. Again, in fact, they use this technology because I mentioned one part of the DNA traits that is a remarkable molecule that you can do everything, and the DNA still has the information that can be transcribed later on, no problem. So if you work with protein, you need to have a cold temperature to work with. So you work with RNA, you need to have a specific room, otherwise you'll lose your DNA. If you have your DNA clean, you can work everywhere, in any place that is possible. And this is, and so this is very simple to work with this molecule. And it's very simple to change. Today, we have many techniques to change. Even in the past, they used to work with mutant agents, like chemical or physical agents to change it. So it's simple, it's cheap, but the third maybe the third issue related to these two is that the people think that a change in DNA will solve the trouble, because this is the language of the science currently. If you open the main magazines like Science, Nature, DNA is still there. And I guess there is another factor that even though epigenetic was proposed as as a control of the genetics, control in the sense that will modulate the gene expression, not control totally, but in the way to modulate the gene expression, was proposed in 1939, I guess, by an England guy, Waddington. But you know, it's 80 years in the role, and few people talk about that, and few geneticists teach this in class. So, if you learn just these two first topics, that is how remarkable is DNA as a strong molecule, and how easy to change, people start to believe that and some of them is because they don't know, they didn't face it, they were not exposed to the holistic approach to solve an issue. So this is my opinion. I have been in touch with the philosopher for a long time as well. And something that I learned with these guys is that so if I am a lab leader, I can choose the research line, and my students can go through in this. So if there is different kinds of research or science, but there is this majority one that is called sound science, that is science developed by some people who have the interest to sell, and people will use the products or service of this science research. And the other type of science is a science that is regarding to the needs of the population itself or the troubles itself. So you will not develop a product or a service for people use. No, you develop a solution for a trouble. Some scientists call this like a contextualized science in the context of the people. So these guys that try to develop solution for a trouble from the, say, so I have the solution, I'll do that to solve the trouble. This is not a contest, not in the context of the trouble. So it's the trouble they think in this way. And they will apply in several places of the world. And maybe it's not the main demand of the community or the trouble itself. So I always try to work in the context of the environment and the people. I have been doing other kind of research, but I work with farmers, with participatory breeding. So when you have this principle in mind, always you will develop science that will be useful for the people. However, I'm doing this kind of research with GMOs and gene edition because I'm looking for the risk of these technologies. In fact, it's my way to say I will contribute to the society to debate this issue and find a solution. So I will not impose the products of my research, our research with the students, but we provide data for people to discuss about the issue. Because I think it's the main trouble for the scientists is that I think we have the role to provide information, reliable information. But it's the society that should decide if they use or not that issue. In the last, since the this DNA revolution about knowledge in the 70s, I guess this role of the science, it is the scientist was broken apart. So the scientists now believe that they have the solution. And also the media think that we scientists, we should give the final word like it happens with the vaccine and everything else. So usually I don't give my opinion to the media because I'm not supposed to. Usually when I have interview with the media people, I talk about the troubles. So like last week I say, well, we are using too much pesticides. So these pesticides cause harm. How we solve this? So we need to join as a society. So most of the scientists that I know, they believe that they have this missionary role to solve the problems. It's not the trouble must the society to be solved, not the scientists alone. So this is one of the reasons that we use some techniques or we design some research lines that we produce some product, service that is in our mind. But so our mind not necessarily is the main, which is in our mind is not necessarily the best solution for a trouble. So I think I think the society must define again the role of scientists. We should provide reliable data regarding one trouble, one demand, in the more wide picture or vision than in taking the holistic approach, instead to make a recommendation for just based on a single part or a single issue of a big trouble. Yeah, this is the you are right. This is the scientific arrogance of the biotech people. I never met arrogant people in other scientific areas. Well, there are, but but the proportion of arrogant people in biotech is over any kind of science. You don't find this too much in chemistry or in physics maybe, but in chemistry and other areas. It's amazing that this behavior is because these people, again, they are based on the central dogma. Yeah, there is, regarding to that, when these guys try to say that, oh, so I'm illuminate guy, so I develop a solution for a trouble, so I spend time, money, so then people should use. And they say, I don't have money to do all the risk analysis that could be required for releasing this. So then they put a kind of limitation to do the risk assessment analysis because it costs money. And this happens with GMO. This happens with vaccine. Now, do you see what Pfizer did? No. Government, if a government buy my vaccine, government assume all the risks regarding to the adverse effect of this vaccine. Again, they are the same guys that use biotechnology to develop a product, but they don't want to carry on any risk analysis because it costs money. I don't have the solution. we try to dialogue with some authorities in our government, and we are fewer in comparison with the biotech people, the number and the power that these guys were able to reach in the past 20, 30 years. So if we sit together with, I have experience like to sit at the side of one biotech that developed a GMO in Brazil, and so usually in our case we had more recognition for the issues that we brought about in like 20 years ago. We have a good recognition for our ideas regarding to the trouble, the risk assessment needs, and however, we had I should say modification or replacement of people that has concern about the risks and of this new technology. So these guys were replaced by new ones. And we have 3 or 4 other governments that every 4 years, every new government less care about these problems were replaced by the good care that was in place 20 years ago. So in our case now, most of our under law regulation became flexible, more flexible. So I give you one example. In our first regulations for testing a new GMO for allergy should be done in vitro and in vivo. Now is the developer can do in silico, just comparing the. So in my opinion, based in our government, they care about the risk of these new products is very small in comparison with the past governments. So I should say that I think remain for us the group of the people that are concerned about the adverse effect of the new technologies. So we should get together as society or organization and restart again a kind of dialogue with government. And could be a good idea because, when research are in place, usually will be a fight between research. So if you get organized in a society, an informed society, I should say the first issue is move to spend more time with society organizations, non-governmental organizations, and the people must be informed. It's not easy, it's not simple, takes time, because some issues are a little bit complicated, but in my opinion, is the last option that remains to us to maybe cause some sensibility to governmental agencies and or ministries or something like that. I don't believe that the scientific organization has power to do, because in our country, most of the scientific society became addicted to the biotechnology. So most of the people use this technique. So I think it remains to the society, the civil organizations, to provide some dialogue or some politic incidents in government agencies and so on. This is, so information, that we have good ones right now, in organization.